Welcome everybody. We're going to learn about the science of spider webs with Dr. Stellwagen here, and I'm going to turn it over to her. Thank you so much for joining us this evening.
Awesome. Thanks so much for inviting me. I'm super excited to talk about spiders. Halloween is my favorite season because all year long, spiders are these creatures that we kind of are, icked out about. But then Halloween rolls around, everybody loves them. They decorate their houses with them, they talk about them. And I just think it's a wonderful time of year. Not all creepy crawlies get their own holidays. So I love it. This Halloween happens to be a hunter's moon, so the second full moon in October, which hasn't happened in a really long time, and it's on a Saturday. So I feel super sad about coronavirus and what a wonderful holiday it was going to be, but we're still going to make the most of it. And I'm glad that you're all here to learn about spiders, especially since I think if coronavirus wouldn't have happened, these things wouldn't be live on Zoom, and so a lot of you probably wouldn't be able to participate. So making some lemonade. All right, so I will share my screen here. Let me see. Share screen, Microsoft PowerPoint. I mentioned earlier, I have a sleeping baby behind me behind this wall. If she starts crying, my husband will grab her. So just try to bear with us, however long that takes. It shouldn't be long, and I don't think she'll wake up, but so today I'm going to talk about spider webs and spider silk, particularly the glue that covers the spiral parts of orb webs. For those of you that have kids, some of this will be totally graspable, especially at the beginning. I will get into a little bit of nitty-gritty science. I tried to tailor this to an audience that doesn't, this isn't a research talk, but it does have some research in it so that you can kind of see how I go about studying spider webs and some of the projects that I've worked on. First, I'll talk about some of my PhD work, which involves some mechanical testing of silk. And then I will talk about some genetics work that I've done. And at the beginning of both of those talks, it's more broad. So if we get into the deeper stuff of the biomechanics, if you wait past that kind of deeper part, I'll go back a little bit higher level again for the genetic stuff, and then we'll dive down deep again. So at some point I might lose some of you, but for the people that are more interested in the sciencey stuff of it, we'll hopefully have some for everybody. So just a few different types of webs. Of course, we have the big, beautiful orb web, which is something that I've done most of my research on, although I'm starting to branch into other types of webs. This is beautiful web. It's hard to catch them in the sun like this. This is another type of spider web, and this is called a gumfoot web, and they think of it as a modified orb web. So the orb web came first, and then evolutionarily it was reduced or modified into gumfoot webs. The types of spiders that make these webs are widow spiders and their relatives. So the way that they make these this is the tangle and the little spider in here. And then they drop these lines down from the tangle of their web, and they very loosely attach them to the ground. So if you see a zoomed in picture, you see these little glue droplets just right there at the very tip, and you can kind of see them in this picture too. So what happens is an insect will come along, it'll bump into this strand, and because it's so loosely attached, it will pop off the substrate, and because it's gluey, the insect will be stuck on the strand, and the spider will just come reel them in kind of like a fish. So that's another type of spider web that are around, but they're just harder to see and it's harder to notice for sure. Another type of spider that are definitely around, and certainly around Maryland, because it's the only place that I've seen them, though they exist all over the United States and other places in the world. These are called bolus spiders, and it's one that I'm particularly fascinated with right now. The way that these spiders catch prey, and they typically prey on moths, which they mimic the pheromones. So when the moths are mating, the male moths will make a pheromone and attract the females in. Or it might be vice versa. One of them makes a pheromone that attracts the opposite sex. This bolus spider makes a pheromone mimic that attracts the moths into them. And what they do is they wait with their little sticky glue ball here at the end of a strand of silk. And when the moth gets close, they actually take that and they whip it around and they chuck it at the moths and catch them out of the air. It's absolutely insane that the way that they do this. These spiders are big. They're about the size of an acorn or a little bit bigger. And you've probably never seen one. And they are incredibly cryptic. So they look like bird poo when they're sitting on a leaf during the day. They don't make webs, so you can't find their webs. I had to contact a naturalist in Maryland in order to find these spiders for my research, and she knew of two. And I went out hunting for these spiders probably 5 or 6 times, couldn't find any, took my husband and he found one, which was really irritating. So I still personally haven't found one, but I have seen them and collected them because other people have helped me find them. But they're not necessarily rare, they're just really hard to find. If you have a pen and paper, or you can just remember this, there's a really great video on YouTube. If you just type in redback spider BBC Earth, it shows an example of the previous spider web that I just showed you, the gumfoots, and then right after it is the boltless spider. So they're both in there. It's a part of the David Attenborough narrated Life of the Undergrowth. It's really great. It's about 6 minutes long. So if you want to see examples, that video is really, really great. Okay. So spiders make a lot of different types of silk, particularly these orb weavers make 7 different kinds. And they all have different material properties. So in this case, you have the major ampulate silk. This is the typical silk that a lot of people have been working on trying to mimic and make a product out of. And this is what's responsible for the frame of an orb web. Then you have minor ampulate silk. They use this for the spokes of the wheel of the orb web. A ciniform silk. So if you've seen the writing spiders, the big black and yellow writing spiders in your yard, in your gardens, the stabilimentum, the little writing part, is made out of ciniform silk, and also what they use to wrap up their prey, if you've ever seen a spider feeding. Piriform silk, so this is what they use to stitch all of these different silks together. And then tubuliform silk. So this is the silk that they use to wrap their egg sacs. The last 2 types of silk are the types of silk that I've really been studying. And there's 2 different kinds. There's the flagelliform silk here that you can see. This is under a microscope. And this is the spirally part of the web. And supporting this on the silk is this glue droplet. So the glue droplets are supported by the flagelliform silk. Now this aggregate silk, as it's called, doesn't form a fiber anymore. So most silks you think of as this long strand, kind of like a thread. In this case, the aggregate silk is a liquid silk. So all silks are proteins within the spider, a liquid protein. So they call it a dope, a liquid dope inside the spider. And then as it's coming out, the spiders pull that liquid dope into a strand. This silk, however, is a liquid inside the spider, and it's extruded as a liquid. So it doesn't go through that transformation. Evolutionarily, it comes from solid silk. So what they think, or what arachnologists think what happened was that a solid silk turned into the liquid silk. It didn't kind of evolve on its own. Okay, so where does the silk come from? So at the very end of the spider, so this is a spider that's in its web, it's head down, and we're looking ventrally, so the belly side. And then right here are the spinnerets of the spider. So if you zoom in, you can see that there's a few pairs of spinnerets here. Now, if you zoom in even closer, under a microscope, this is what you see. So you have a few different pairs of spinnerets. These 2 you couldn't see in the previous picture because they were covered. The pair of spinnerets that I'm really interested in are these posterior lateral spinnerets. So if you zoom in even closer, you can see all of these little spigots. So this is what the silk actually comes out of. So each one of these is an individual strand of silk. And in this case, all of these little spigots make prey wrapping silk. So the aciniform. And so that's when you see all of these bunch of little fibrils coming out at the same time. But what I'm really interested in are this little triad of spigots, and this is where that flagelliform fiber and then the accompanying glue come from. So you can zoom in even closer on these little spigots here, and you can see there's this one in the middle. So this is where the fiber comes from. And then flanking that are this pair of silk ducts that extrude the glue onto the fiber. Okay, so when I'm doing my studies, I have to dissect spiders. And as I mentioned, the silk is actually a dope, so a liquid dope inside of the spider. And so where do they keep that? And this is kind of a messy picture, so don't think too hard about it. But I just want to point out when I'm pulling the spiders open and trying to find the specific glands that I need to do some DNA studies for, this is kind of what I see. And so this is the duct from a gland leading down in the spinnerets, which would be below all this mess. And then this little balloon-shaped thing is a gland. So this is like kind of like a balloon. So if you think about a balloon full of water, and then the nozzle of the balloon is where the silk would come out. And so this is a very full gland of silk protein right here. These little berry-shaped ones up here, that's the aciniform. So remember, there's all those little spigots. There's a whole bunch of these little ones, and each one leads to one of those spigots. Here's another gland. So all of these are different ducts and glands that are all kind of jumbled in here. This is kind of a more clean picture after I pulled out all the glands and separated them. The ones that I'm really interested in are these glands here. So this one up here and this one down here are the tubuliforms. This is where the egg sac silk is made. They have 3 pairs of those. But this is the aggregate gland. So this is where the glue is actually made. And then this is the gland where the silk fiber comes from that supports that glue. When you're trying to pull out these glands, most of these are pretty easy to see and they're very distinct, but the cloudiness of these aggregate glands just kind of explodes in the spider, and you kind of have to peel it off of all the other organs and off of all the other glands. It's a real challenge, and it's hard to get them completely intact. I don't even know if you can get them completely intact because you're indefinitely going to leave some little bits behind, but you can get enough of it to do some work with for sure. Okay, so droplet formation. So when we're thinking about these strands of silks with these droplets on them, how does that happen? Are the spiders putting them individually on the strands? And the answer to that is no. What's happening is, as the silk strand is being pulled and the glue is being kind of dumped onto the strand, the glue is really hygroscopic. And what that means is it has all of these salts and low molecular weight compounds that pull in water from the atmosphere. And they're so hygroscopic, they do this before your very eyes. So if you have a droplet in low humidity, in this case, this is like 20%, which does our environment even get down to 20% naturally? But if you have it in a little chamber or a temperature or a humidity controlled room and you get the humidity down and then you just breathe on the droplet, it will swell instantly. So this is the same droplet under different humidities. And so they swell instantly and they swell quickly, or they swell into pretty large sizes. So what happens is the spider is extruding the glue onto these strands, and then it rapidly absorbs all this moisture, and then just natural surface tension breaks them into these droplets. And I've seen pretty big spiders making their webs and doing this. So you can see them extruding the glue into like kind of a cylinder as it's coming out, and then it just kind of starts breaking up as time goes on and the glue starts to swell. So what does it look like on the inside? So this is a single droplet of glue, like the one that you see above here. And we've taken a microscope slide, just a glass like cover slip, and put it on top of the droplet so that it's squished on there. And what you can see is the supporting fiber, the flagelliform fiber. Now remember, when we looked at the spinnerets, there's actually pairs of spinnerets. So every time you see a silk strand, it's actually 2 strands because the spinnerets come in pairs, the ducts come in pairs. So all silks are actually paired. So in this case, you can actually see both fibers at the same time. And then the glue itself has 2 kind of distinct layers. First, it has that aqueous coating. This is where all the salts and stuff live that makes it absorb water. And then it has this central part. This is the glycoprotein. So this is the part that makes it sticky. So the water and stuff keeps it nice and plasticized, keeps it nice and gooey. And then this is the part that actually sticks. So here is a cool picture from when I was doing my PhD at Virginia Tech with my advisor, Brent Opel. He had taken pictures of lots of droplets. He did tons of droplet extension studies, which I'll get into in just a minute. And so he had taken really cool pictures of all the droplets. So this is all at the same scale. So obviously some spiders are bigger than others, and so they make larger droplets. These are Gaiape species. These are those garden spiders. We have 2 species of those. Varicosa, this is one that you may have seen in the woods. It has a little triangle on its abdomen. It's a daytime orb weaver. They hang upside right in their web, and their triangle on their abdomen is either yellow or white. Microthena gracilis. This is one you've all probably seen. It's spiky. It's like black and white. It's kind of small, and it's very spiny abdomen. And just a couple of others. So just a really neat picture of all the different droplets from the different spiders. So I want to mention before I talk about my droplet studies that I did projects looking at individual droplets, but really, when you think of a spider web catching an insect, they're not intersecting one droplet at a time. They're getting stuck to a bunch of them. And some work has shown that that they really work like a suspension bridge. So when you hit a bunch of droplets at once, they all stick and they combine all of their forces to really keep an insect stuck in a spider web. Okay, so the first project I'll talk about is one from my PhD. And in order to study spider glue, you have to collect droplets and webs from the field. So what we would do is we would take these sticky rings. So this is just an aluminum ring that has double-sided sticky tape on it. You go find your web, you kind of shoo the spider out. We collected the webs right after they're made really early in the morning. So I would go out in ridiculous hours, or at least it felt like it for a grad student, like 4 or 5 o'clock in the morning to get really fresh webs. And then I would collect them and then go immediately back to sleep. But we would take these aluminum rings, and you basically you find the web, and they're 2D, of course, and you take the ring and you just push it up against the web gently. You can kind of squish the silk down around the sides on the tape. And then you take a scissor and you cut around the web. So what you end up with is a piece of the spider web at its native tension on this aluminum ring that you can now take back to the lab. Because we want to test these pieces of silk in their natural state. And the only way to do that is to take the webs back without disturbing them. When we get them back to the lab, we would take some forceps and touch it real gently to one of the spiral pieces. And then we would burn using like a soldering iron tip with a really hot, super hot tip, burn out either side. And then so that's still at its native tension. And then we would touch it down onto these supports that were glued onto a glass microscope slide. So now we have pieces of spider silk suspended at their native tension on a microscope slide that now we can manipulate. Okay, so what we would do with those little glass slides is we would stick them into this little chamber that's sitting underneath a microscope. So here's the little chamber, and in here you can see a microscope slide. And this chamber allowed us to control the humidity and the temperature, which is what we need to do when we want to test these at, if we're testing the humidity or we're testing the temperature, we need to be able to control that. And then in the side, it's a little probe that you can push in and intersect with one single droplet. So the camera flips the image. So the probe in this picture looks like it's coming in from the opposite side, but it's actually coming in on this side and touching a droplet right about here. When we're ready to test the droplet, we turn on a motor, the whole stage starts to move. So the probe starts moving away from the suspended droplet, and you start to see each individual droplet stretch. And I'll show you a couple of videos of those soon. Another thing that we did was to measure the amount of glycoprotein. And I won't talk too much about these studies, but just so you know that it's out there, we were able to look at the different glycoprotein amounts at different places in the web. So spirals toward the center or spirals toward the outside, do they have the same amount of protein, or are there different amounts of protein? So there was a little rig here on the inside of this chamber, which isn't pictured, which held a glass slide in place. So we could take a picture of the droplet as it was suspended. And then a little magnetic clasp allowed us to drop a microscope slide onto the droplets so that we wouldn't have to take the cover off and mess up our temperature and humidity. So Dr. Opel had a little like magnet thing here that allowed us to do that, which was pretty cool. So we would flatten a bunch of droplets too. We could take a picture of them suspended and then drop the cover slip and then take a picture of them after they were flat. So the way that we measured a stretching droplet was in a couple phases. So it starts when a probe is intersected with a droplet, but then it had the line, so the flagelliform fiber that's supporting the droplet is at 180 degrees. So basically straight up and down. Then you have, so this starts the pre-extension phase. So the contacted droplet isn't stretching yet, but you start to see some tension on the silk. And then this phase ends, this pre-extension phase ends when the droplet starts to stretch. So this is at the very start of the stretch and then all the way to the end when either the droplet completely pops off of the probe, or in other cases, the droplet is so wet that it will actually still be attached to the thread, but the thread will be back to 180 degrees. And you'll see an example of that at the end. So there's a different way that we release technically, but this stretching part is called the droplet extension phase. So here is a video of this. Hopefully you can all see that. So right now we're in pre-extension, nothing stretching, but there's definitely tension on the thread. Then the droplet starts to stretch. And it stretches for quite some time. And eventually it will pop off. So remember, this glue is on all these orb webs that you see around your house. You took a piece, you put it under a microscope. This is what it would look like if you start stretching it under a microscope. Okay. So once we have videos of all of these droplets stretching, we actually start to take pictures at different parts of this droplet extension phase, and we measure this angle. And this is just a way for us to try and get at how much energy these droplets are actually absorbing. So we measure this angle. So you would think if this is a tighter angle, you're gonna have a lot more force than if it's not quite as acute. Okay, so then ultimately, here's what we're getting at in the end. On the x-axis here is time. So this is just how long did these droplets stretch. And then on the y-axis here is that angle that I just talked about. So how intense was that angle? So the higher it goes, the more energy that these droplets are absorbing. And so when you combine those two, what you get is this area underneath the curve. And that's basically a measure of how much energy the droplet actually was able to dissipate. So this is what brings me to an important point. So this little shaded area is kind of like a measure of toughness. And this is a buzzword that you hear about spider silk all the time. And I feel like I have to mention this because spider silk it gets talked about in the media a lot, but I feel like it's misrepresented in a lot of ways. Spider silk is very tough, meaning that it can absorb a lot of energy. But as I just talked about, it's a combination of how far something stretches and then how stiff or how much energy it can basically absorb during that stretching. So in the case of Kevlar, which we hear about all the time as bulletproof vests, it's super duper stiff. Like you can't stretch Kevlar, like it's harder than rocks. And so because it's so stiff on the y-axis here, you got, way, way up. And then if you're measuring all of the shaded area under that curve, you get quite a lot of toughness, right? Because it's not stretching very far. But here, spider silk stretches really, really, really far. It's a super stretchy material, but it's not that stiff. But if you look at these two materials and you measure this areas under these curves, they're similar. Like they're absorbing the same amount of energy, but they're doing it in different ways. So if you had a bulletproof vest made out of spider silk, which probably you've all heard, oh, we're going to make spider silk bulletproof vests. I mean, yes, it could probably stop a bullet, but the bullet would like already be all the way through your body because it's stretchy, like it stretches to absorb this energy. So, people don't understand, like you have to get really down and understand the different pieces of the way you're measuring these materials before it starts to make sense of why spider silk is so great at absorbing energy, and that's because it stretches. So when everyone says, if anyone says to you, oh, bulletproof vest, be like, no, no, toughness is measured by both stiffness and extensibility, and both of those properties are important. Okay, so taking all of these techniques that I just showed you, one of my PhD projects was to look at how temperature influences these droplets. So we knew that humidity affects them a lot, and I showed you those pictures of the humidity changing. And basically what happens with the humidity is you get an increase in volume, of course. You get an increase in extensibility, so it stretches further, which is not necessarily a good thing. And it increases the amount of energy that it can absorb depending on the species. So if you have a species that functions really well and mid-range humidities, like 50% humidity, if you put it in too high humidity, it gets really wet and over lubricated and it doesn't absorb as much energy. If we're talking about nighttime orb weavers that function in really high humidities, they absorb more and more energy the more humid it gets. So this adhesion, or the energy it takes to pull a droplet off, depends on the species and where they evolved to catch their prey. So when I started, I wanted to know, well, temperature is also fluctuating during the day, just like humidity. So this is 24 hours. You have lower temperatures in the evening, they go up during the day and then back down. And so I wanted to know, like humidity fluctuates and affects these droplets, does temperature do the same thing? One thing you need to know about these glue droplets is they're a viscoelastic solid. The way I describe this to people is honey is a viscous substance. So you have a jar of honey, you put it in the fridge, and you have a jar of honey and you put it in the microwave. There's spoons in those honey jars. The one that you put in the fridge, it's going to be a lot harder to pull the spoon out of the cold honey. And that's something that we can all imagine. And so that's kind of what I was hypothesizing here, is that when you lower the temperature, it's going to make the spider glue more tacky, and then it's going to make it harder to pull off than if you put them in a warm environment. They're going to get really thin, and it's going to be a lot harder or a lot easier to pull them off a surface. So I tried a couple of different things. So for the spiders that I was looking at, these were those big Argiope species, and they're catching their prey in mid-range humidities, 50%, and when it's really hot outside. So late summer, boiling hot, middle of the day, pretty dry. So I tried that condition. I also tried a typical morning, and this isn't normally when they're catching prey, but they can catch prey. They're in their webs after they've been made. So I wanted to see, okay, well, if they are functioning the best in the afternoon, but then you switch those two conditions in the morning, do they compensate for each other? So you have low humidity in the afternoon, which makes it kind of tacky, but you have high temperature, which makes it thin. Well, in the morning, you would get the opposite. Now you have really high humidity, which makes it thin, but does the lower temperature kind of account for that by making it tacky again? So this was kind of like a flip-flop of conditions. Does that kind of stabilize how the glue functions? And then I just threw in a day of like, this is a rainy afternoon. So this is temperature fluctuating over many days and humidity fluctuating over many days. And this is like a rainy afternoon that's misty, but still hot. And so I was thinking, okay, this would be like a double whammy of bad news for this glue. It's really humid, so it's gonna make it too liquidy, and then it's hot, so that's making it liquidy. So this is just gonna fail, probably, hopefully. This is my hypothesis. Okay, so this is the general results. So remember those charts that I showed you of how much energy something can absorb is the area underneath these curves. So these are also combined. So the afternoon condition here is the darkest one, and it's all three of these combined. So just think of all of this as the amount of energy it can absorb. And it definitely works the best when the spiders are foraging. So it absorbs the most energy. In the morning, it definitely can't absorb as much energy, and that's because humidity affects the droplets more. It's more important than temperature, but temperature definitely does stabilize it to some degree because, oops, if you look at the hot and humid condition, that one absorbs even less. So best in the afternoon when they're catching their prey, makes sense. Morning, you lose a little bit of functionality because humidity is really, really important. And then when you give them hot and humid, they absorb even less. Okay, just to drive this point home, make it super clear, here's some videos. First, there's all three are gonna play at the same time. So these droplets are gonna start stretching at the same time. Keep your eye on this bottom one. So this is unusual afternoon, hot and humid. This is the double whammy of bad news. Then this is the morning, cool and humid. And then this is afternoon, hot and dry. So this one's gonna go really fast, and then this one will go next, and then this one last. So you can really see the difference of what these conditions do to the droplets. Okay, so the videos are playing. So pre-extension phase, this one's gonna start to stretch, and then boom, like this is back to 180. There's no more tension on the thread. So even though it's still connected, basically it's done. The morning, cool, humid, once it starts to release, it goes a little bit slower, but now it's already back to almost no tension on the thread again. Now here's afternoon, hot and dry. We haven't even started stretching yet, and then when we finally do, it sticks really hard, it sticks really long, the tension on the thread is strong, and then it finally releases. So you can see that these conditions do different things, and they, humidity is definitely more important. But it was a really fun study to do and just kind of get at how conditions really affect nature in these little subtle ways.
Sarah,
Okay.
Sarah, couple
Sure.
questions, right, going on what you were just talking about. One is Barbara wanted to know, when you were transporting the webs on the metal hoop, how could you transport it without damaging it?
So the metal hoop had double-sided tape on it, and so we would put them in a box. Basically, we would like put the taped ring up against a web, kind of stick the edges of the web down onto the tape, cut it out of the rest of the web, and then they had like little dividers on them, so we would stack them in boxes, and each one had a space like between them, so the different webs wouldn't touch each other. So we put them in a box essentially and transport them to the lab. Does that answer the question?
Yeah, and then Carolyn was interested in, it goes along with this, is do spiders just make their web? Is it in the morning or afternoon? Is it species specific? And how long does a web traditionally need to last?
Those are great questions. So it is species specific. So a nocturnal spider will make its web in early evening. You've probably seen them on your porch before making their webs. This particular species from this study is an afternoon orb weaver, so it's catching, or a daytime orb weaver. They make their webs early in the morning, so 4, 5, 6 o'clock in the morning, and they remake them almost every day. So a spider will take down the spirally part, the rest of it leaves up, but it recycles the spirally part because it has all those salts and everything in the glue. So it'll actually ingest anything, all of that part, and then some of it can recycle and some of it can't, but it will ingest and recycle as much as it can and then rebuild the next day. So the webs get damaged, they get dust and debris in them. Sometimes they'll keep them up for a couple days, but usually it's every day or every other day they'll remake their webs.
And then what is the adaptive value of this being able to stretch more than under certain conditions than others? What does that benefit for them?
So it has a lot to do with prey type. So you want something something that's too brittle or too stiff can break really easy. For some species, that's good if they're catching really slow prey. But if you're catching like a really fast grasshopper, like you want it to stretch and then they bring the grasshopper back. So it definitely has to do with what they're catching and how fast those things are going.
And is the glycoprotein the same in all the species?
Well, that's a great question. And I'll talk about the genetics of them next. The short answer is they're similar because they're related. But we honestly don't know because it's really hard to sequence spider silks, and we don't have that many sequences, which is kind of crazy considering how popular spider silk is in general in the world, both colloquially and scientifically. But I'll talk a little bit more about that.
Okay, go on then.
Awesome. Do we have like a time limit? Because
We're, we got on a little bit late, so we can go and people can drop off as they want to, because this is important. So go as long as you want to.
Okay. So the next thing I'm going to talk about, this is the second big part of the talk. If you have little kids and stuff, once we get through kind of the more general stuff, they might start to get a little bored, or if you find the genetics part of stuff a little bit boring, then that would probably be a good time to head out. But hopefully I'll be able to put some of this stuff in context where you'll be able to kind of follow along towards the end. So spider glue genes, I went for my PhD working at Virginia Tech doing these probing of the droplets. And then I started at the Army Research Lab, which is in Adelphi, Maryland, just north of DC. And they were hiring someone to do some microbiology, so working with cells and bacteria and stuff. And I had no molecular biology skills. I didn't know anything about DNA sequencing, but I know as a biologist going forward, it's the new, like you have to do some molecular biology to work to be able to continue in this day and age. And I needed those skills. So I said, I'll do whatever you want. Just teach me the magical ways of sequencing and molecular biology. And so I did some projects for them. And then this was a project that I really took forward on my own as like my first independent scientific experiment. And what I really wanted to know was, what is the genetic sequence of this glue? Like, we know how it functions as like a real thing. We know that it's a protein. You saw the protein in those pictures, but like, what is the genetic sequence that encodes for those proteins? So here's that saga, which was long and arduous, but ultimately successful. So spider silk genes are called spidroins, which just is a combination of spider and fibroins. Fibroins refers to all types of silk. So silkworm silk is a fibroin. And we just stuck spider at the beginning and called them spidroins. They're really large, and what does large mean? I'll tell you about that. And they're really, really repetitive. So most proteins have an N-terminal domain, which is kind of unique, and a C-terminal domain. Those are just like the caps and the end of a sequence, essentially. And then for silks, they have another part of their sequence which just is repeated over and over and over and over and over again. And this makes them kind of complicated. So what does big mean? We're talking about DNA sequences and then the proteins that those DNA sequences encode for. So that average size gene is about 1,350 base pairs, so DNA molecules long. The largest in E. coli, which is our favorite laboratory bacteria that we mess with, its largest gene is about 4,000, a little over 4,500 bases. The largest known DNA sequence is actually encodes for the protein of your muscles. So that's called titin, that protein. And it's anywhere from 80 to 100,000 bases, which is really big. But the way that those genes work is you have a little bit of DNA that encodes for the protein, and then you have a bunch of DNA that doesn't. And then you have a bunch of DNA that encodes and then a bunch that doesn't. So introns and exons. In the case of spider silks, most of them don't have introns. So the bit of DNA in the middle of genes that doesn't really do anything or does something else, but isn't protein encoding. In the case of spider silks, they're anywhere from 5,000 to 20,000 bases. So they're really big when you're thinking about an average sized gene, much bigger than the largest of a bacteria. Okay, so don't look at this too hard, you'll go blind. But this is just an example, a couple examples of spider silks and the proteins that make up the silk essentially. So this is the N-terminal domain and the C-terminal domain in these colors, and these are really unique sequence. And then these amino acids are just repeated over and over and over and over again. And this is encoded by almost 10,000 bases. Similar story here, almost 10,000, many, many, many repeats. So this is tubuliform silk. This is the silk that spiders used to wrap their egg sacs. And this is from an Argype species that we don't have here, but is in different parts of the world. So just a different type of garden spider. Here's another one, a cynniform. So this is the prey wrapping silk. Many, many repeats, 19 repeats. They're almost always exactly the same. For the major ampulate silk, the repeats can vary a little bit, but not much. And they're just repeated. It's incredible how consistent this is and why nature supports that. You know, if it wasn't important, it wouldn't be there. Okay, so this is a chart showing this is the species of spider, and this is all comprehensive list. I was when I made this a couple years ago, and it hasn't expanded very much at all, if any. This is a comprehensive list of complete spider silk sequences, of which there's about 20. So this is how long they are. So 5,000 to 20,000, which was the largest at the time. It kind of blows my mind to think about how important silk is scientifically and just in our culture. And then to go, when I started doing this project, to think about, oh, there's only 20, we only know 20 sequences. Like, that's so weird. But we know a lot of fragments, it turns out, because sequencing these crazy repetitive genes is really hard. And I didn't know that. So I was like, I'm gonna go sequence this spider glue gene. It's gonna be really easy, because like, why not? Because I didn't know anything about molecular biology at that point. And boy, did I learn a lot. Okay, so to bring everyone up to speed of why this is so hard. If you're not a geneticist or you don't deal with DNA sequences, hopefully this will give you kind of like a grasp of how we do the sequencing and why this is hard. So imagine this sentence: the quick brown fox jumps over the lazy dog is a DNA sequence, and each one of these letters is the base of DNA. When you're doing gene discovery, you don't know this sentence, and I'm giving it to you for the sake of the example so you know it. But what really happens when you're doing discovery is you take a bunch of copies of the sentence, which you don't know what it is, you chop it up into a bunch of little bits, you throw it on the DNA sequencing machine. Out the other side is you get little pieces. And this is the technology up until a few years ago. This revolutionized the sequencing of DNA over the past 15 years. This is a really important advancement, but it's hard for repetitive sequences, and you'll see why. So you take your sentence, you have tons of copies of it, chop it up, you get all these little pieces. As long as the pieces are long enough, it's kind of like a puzzle, and you just stack them on top of each other, and you can reform whatever the sentence was or whatever piece of DNA you have was. So this is very kind of like a simple puzzle, and that's exactly what DNA sequencing was. You have a unique sequence. Oh, it's here in this one. You stack them on top of each other. Oh, that's what it was. Repetitive sequences. Imagine you have a sequence, the quick brown fox jumps over the lazy dog. Well, you can figure out the unique parts. So remember these termini on either side. You have really unique sequence, so you can stack those sequences and figure out what this part is. You can stack sequences over here and figure out what this part is. But what about the middle? Maybe you know what the middle part is. It's jumps. That's our repetitive unit. We know what the repeat is, but how many of them are there? Is there 2? Are there 10? Is there something in the middle? So this is really the problem with sequencing spider silk. Is they're really big, they're really repetitive, and these the sequencing technology that we have the pieces of DNA that you get out aren't big enough to go all the way across. So you need to go from the unique portion of a gene all the way across the repeats into the other unique portion in one single go. Like you can't have pieces of it. It has to be one piece in the machine out the other side. And before, just a few years ago, we didn't have this technology. And that's why, without going through some really incredibly difficult methodologies that only a very few labs specialize in, could you do something like this? Okay, so here's where I was when I was trying to figure out this gene. In 2009, before I had even gotten to the Army, there was a group that came out and they're basically like, oh, we sequenced the silk gene, the glue genes. There's 2 of them. We're calling them aggregate spider glue 1 and aggregate spider glue 2. And here they are. Oh, by the way, here's all these ideas that we've thought about and we're going to patent them. And so this is the state of this for a few years. So they were saying that the first gene was about 1,000 bases and the other gene was 2,000 bases. And they were like, this is complete. Obviously, we've done it. You're welcome. 2016, I'm at the Army. A paper comes out that basically says that the first gene from that other group is not a spider glue gene. It's everywhere in the body. It's expressed everywhere in the body, not just in the glue glands, which is what you would expect, that the glue gene is only going to be expressed where the glue is made. So this is not a glue gene. We don't know what this is. The second one was a glue gene, but it had a little extra piece of DNA in there that wasn't actually part of the gene. So they had a mistake. And so what this new group discovered was yes, that was a glue gene. It had a little mistake and they also added to it. So now it's almost 3,000 bases long. So this is where we were when we started. I had about 3,000 bases and only one that we know of. So the first thing I tried, which is what I just told you doesn't work, but I didn't know what I was doing at the time. I was like, we just got this new Illumina machine. So Illumina is the big company that does all of the short read sequencing. Short reads, as I told you, don't work, but the lab just bought one. I was like, let's try it. Let's like see if we can do it. I don't know. So we ground up some spiders, we threw it on this machine. And of course, it didn't work. I banged my head against the wall thinking, oh, wow, maybe I could just figure this out. And there's like some magical thing here that's going to make it all work. But of course, I just showed you why it doesn't work. So that just doesn't. You can't make it work if it doesn't work. But so we got the unique parts on either end of the gene and we added to the overall length. So before it was around 3,000. Now we know that it's at least 11,000 because the unique part on the upper end of the DNA, so the 5' end of the DNA, 7,000 bases, that's all unique. On the 3' end, so the other side of the gene was 4,000. Now we're up to 11,000 and we're like, okay, well, that's bigger than what we thought was going to happen, although we didn't know it was going to happen, honestly. But we thought, okay, well, the biggest silk gene that we know of is 19,000 bases. We're at 11. Surely it's going to be like less than 19, right? Right? Less than 19. Okay. Then about a year later, the Nepheloclavipes, so these are those big banana spiders that you see in Florida and on the coast, the golden orb weavers, they sequenced a full genome. And I had heard through the arachnology grapevine that they had sequenced the glue gene. I was like, no, like I'm doing this. This is my project. And like, I was like sad that like I've been scooped and no, I hadn't actually done it yet. But it turns out that they had sequenced some other gene, which I didn't even have in my data. I don't know what that other gene is, but they didn't have the main spider glue gene, but they did have the same bits on the ends. And so I knew I was on the right track. At least I was right about something. Okay, the next strategy that we tried was this new technology has started to come out and really take over. And this is long read technology. So before we were using the short little bits, now we're getting technology where you can sequence really, really long pieces of DNA all at once, which is what you need for repetitive parts of genomes. The human genome is still not complete. You've heard that, oh, we completed it in whatever, 2000, whatever. No, it's mostly complete, but these repetitive regions are still plaguing scientists. There's still little bits of the genome that we still haven't quite gotten because of the repetitiveness of these genes. And so now we're getting to the point where finally we can tie up these loose ends. So we decided that we were going to buy this little machine. You know, it fits in the palm of your hand, from this particular company called Oxford Nanopore. It's basically a membrane. You squirt your DNA on there and it gets sucked through this little pore. And as it's going through, it reads it however it reads it. So we tried that. But the first time we tried it, we used RNA, which so you have your DNA in the genome, and then the bits that are important for that particular cell gets transcribed into RNA. And so we tried RNA. RNA is really, really fragile. And so we thought, okay, well, we'll just try this and see what happens. We did get a really big read out of the other side. So about 16,000 bases. And it goes from the 3' end or this side of the DNA molecule all the way up here. So you can never start from this side. You always have to start from this side when we're talking about RNA. But we went all the way up to here, and these are all repeats, all these little blocks, and it died in the repeats. So remember, we got to get all the way from unique sequence all the way over here in one molecule. And we didn't do that because we didn't see this part yet. That was 16,000. We're like, oh God, this was bumping up against what technology for RNA can do. Now we're up to 23,000. Is now the biggest silk gene known. We're like, okay, less than 30, we don't know, maybe. So the last ditch effort that we did, and I'm getting towards the end here, I promise, we did direct DNA sequencing. So that was RNA. Now we're switching to DNA, which is just grind up the spiders, get all the DNA out of all the cells, throw it on there and hope that you get the molecule that you need. We know that we need really big DNA. So what we did is, this is just a fancy gel machine. You squirt your DNA on this side. It has to travel through this gel. And so the bigger pieces are slower. So all of the little pieces get sucked out into this area. And then the bigger pieces are slower. So you can at some point start saying, okay, I'm ready for you to start shunting the really big pieces of DNA over here so you can suck them out. So we got rid of all the little stuff because we don't care about the little stuff because that doesn't help us. So we basically selected for really, really big DNA molecules to put on our machine this time. We did it. It was like a needle in a haystack. This is the full gene. So it has 2 repetitive parts. It has a big repetitive part over here and a big repetitive part over here. Each one of these is around 20,000 bases itself. This is just a graphic representation of the molecules that it took. So we got a piece that was about 8,000 and then another really big piece and then another one. And so we were able to stack them because they were big enough and there was enough unique stuff that we were able to stack the unique parts. It was 42,270 bases long. That is incredibly large. Remember, the biggest silk gene was 20,000. So this is over twice the size of the biggest silk gene, incredibly repetitive. It was a great day when I finally figured that out because I had gone to the computer so many times and not found it, but it was there. Second, there's 2. So I kept seeing in some of my data this one particular sequence popping up over and over again. I was like, what is that? Like, that's not part of the other gene. Like, where is this coming from? There's so many of them. This has to be important. And it's so similar, but yet it's not there. Turns out there's actually another glue gene. And remember when I was showing you the picture of the flagelliform spigot, and then there's two flanking aggregates. So that means there's 2 pairs of aggregate glands. So what we think is going on is that in one pair, there's one gene being expressed, and in the other pair, there's another gene being expressed, which makes sense because every pair of glands has their own gene sequence going on. So we found another one. It wasn't quite as big, about 20,000 bases in this case, but we got both of them. There's 2, and now we have at least one set of representatives from one spider. But now you know why it's so hard to get these genes and why there's only so very few of them. So what I'm really interested in going forward is sequencing more silks, more glues from spiders, more glues from other organisms. Like, other organisms make glues too. The glowworms in Australia and New Zealand that hang their little strands from the ceiling and put glue droplets on their hanging silks, that's glue that they use to catch prey. What does that sequence look like? Like, I'm so curious about that. Bolus spider, I've done some sequencing on the bolus spider. I don't have the full thing because it is ridiculous. Like, you think that these genes are ridiculous. Turns out those bolus spiders are even more ridiculous. So I'm super jazzed about the sequencing side of this because it's just a whole black box of unknowns that people haven't been able to break into until now. Ultimately, I would love to make spider glue, like get with some people that know how to express these things in bacteria. We can make spider silk. So the problem with making spider silk commercially is it's a liquid inside of the spider. We can make the silk protein, but then the spiders pull the silk and it turns from that liquid into a solid. That's the part that's stopping us. So scientists can make buckets of the protein. That's not a problem. It's like, how do we get the protein from this liquid to a solid in a way that's commercially feasible? That's a bottleneck. People can make it in very small amounts. We have synthetic spider silk. We just can't make it in enough to like give you a spider silk, moo or something, which would be amazing, by the way. But the glue is a protein inside of the spider that's a liquid, and then it comes out as a liquid. So it doesn't go through this transition into a fiber. So maybe if we can make the protein and tweak it a little bit, we could just synthetically make buckets of spider glue. I know one of the questions is going to be, what do you use this for if you could make it? I see it as an environmentally friendly pest control. So it evolved to catch insects. You can imagine like spraying it on the side of a barn. The flies and things land, they're stuck. You can just wash it off. It's water soluble. It's environmentally friendly. It's protein. And then you can just reapply. Mosquito nets, filtration. Like if you put it, like had a filter, you could just like dip in some glue and then like catch all the dust. I don't know. These are things other people have told me in the past too. Or you could just play in it. Like, I just like want a bucket of spider glue. I reach my, like, what does spider glue feel like on like a mass scale? Like I'm super interested to figure that out. But you basically, you put your credit card number here and then like, you tell me the humidity and temperature of where you're at, and then I'll send you a bucket of spider glue for your environment. So that's the goal. And that's basically where we're at with spiders and silk and making it and sequencing it and testing it. So these are all the people that have helped me with these projects. So yeah, I'm definitely happy to take anybody's questions.
That's awesome, Sarah. Thank you. And congratulations on unraveling that the sequence and it happened here. We have a couple of questions. Justine was interested because you were working for the Army. What is the Army? Why are they interested in spider silk gene sequencing?
They're not. I mean, this was like basically, if you know anyone that's in graduate school for science and they want a position like their next step for like postdoctoral work, the military and the government generally have pretty good stipends for postdocs. And then also we live in the military industrial complex, so they just like have more money than they reasonably should have money. But like then as a researcher, you can spend that money to do cool projects. And in this case, I was doing a lot of projects for the Army that they needed done, which is totally unrelated, and something that I did, which was great, but I wasn't that interested in. But then they give you some freedom to explore stuff. And I was on a biomaterials team, so it made sense that I was studying a biomaterial. Like you can't just do anything, but like it was a biomaterial that was cool. So it was like within the framework of what that team was working on. But they weren't this was something that I cooked up all on my own. Was like, I want to try this. I want to, sharpen my molecular biology skills and use this kind of to do that.
And you talked, and earlier on, we had a question, but you talked about there's different types of organisms that make glue, and someone was interested in understanding if the silk that a moth uses in a cocoon is similar or different, or how similar or different they are to spiders.
They're very different in the sense that they evolved independently. So there wasn't like a common ancestor that had a silk and then spiders and moths ended up from that common ancestor way down the road, and the silk is the same. Spiders and moths they evolved their own independent silk. So they're not related evolutionarily, but silkworm silk is repetitive. It's a repetitive gene and has a lot of the similar characteristics of spider silks. It's not quite as tough, like it can absorb as much energy, but it's obviously a really wonderful material that we're all familiar with and can do really wonderful things. So they're not evolutionarily related, but they are similar on the genetic level.
They had, we have a question, are there any spiders, this is interesting, that don't make their own web, but use the webs of other spiders?
Yes, there are. The diversity of spiders out there is amazing. There definitely there's one particular spider called Portia, and she crawls in the webs real quietly and like eats the like orb weaving spiders. So she's real sneaky. So the spider eating spiders, there are spiders. If you look at those garden spiders, so the ones that I study that we have all seen, the big black and yellow ones, sometimes they have like a little what's called a dewdrop spider, and they're little silver, tiny little silver spiders that live in the web. They don't harm the big spider, but they catch all the little stuff that the big spider can't eat, like the little gnats and stuff. A big garden spider, it can't. I mean, they can't even manipulate something that small. So the little dewdrop spiders will go collect the little stuff that the big spider doesn't care about. So they're not really parasites because they're not harming the other spiders, but they're kind of like these little hitchhikers essentially on the other spider's web.
We have a general spider. Another general spider question is, can spiders orient themselves? Do they know when they're upside down?
Yeah, they know when they're upside down, upside. So most orb weaving spiders hang head down. There are a few that hang head up, but in general, the orb weaving spiders, especially once you get to a certain size, hang head down because they run faster heading down. And so they also make the bottom part of their web a little bit bigger. So they hang head down because they travel faster, and then they have more area at the bottom. So they can kind of utilize gravity and a larger portion of the web better than, having even amounts, but they can't get to the top quite as fast. So they tend to tailor themselves based on how big they are and the size of their webs.
And we have, when you crush up spiders, where do you get your specimens?
Oh, it's so sad. I love my PhD.
And how are they crushed?
My PhD, I didn't have to kill spiders that I love, but to get the DNA, you have to kill a few, unfortunately. Basically, you take a little test tube and you put first I euthanize the spiders humanely. So I put them in the freezer for about 5 minutes until they're good and knocked out. And then I stick them in a test tube and kind of like a pestle, like from a mortar and pestle, you basically grind them up in some special liquid, which is kind of like detergent. If you've ever done like the strawberry experiment at home where you take detergent and some alcohol and you can extract the DNA from a strawberry. If you haven't done that, it's great fun for kids. If you have kids of a certain age, you extract the strawberry DNA. It's basically a fancy version of that. It's like a laboratory. It's just detergent, and like, we don't make it. You get it from a company, but it's detergent and some salts and stuff. You grind it up, and then you can just go through I'm mixing this liquid with this little bit of liquid. And who knows what's actually going on at a molecular level, but you start with the ground up spider, and at the other end, you end up with a little tube of clear DNA, which I don't know, sometimes I think aliens are controlling us and like there's really nothing in the tube and they just like upload all the data on the screen. Not really, I'm not a conspiracy theorist, but you deal with little tiny clear droplets of stuff all the time. You start to wonder like what's really in here.
So Lindsay wants to know about the repeats in the DNA cycle. So do you think that does the sequence have a purpose if it's repeating like that?
Yes, the repeats influence the structure of the silk. So with the glue, the repeats are a little bit different than the solid silk. So the solid silk's kind of like, depending on the sequence, kind of turn back on each other, like the protein folds up so that some parts are stiff and some parts are stretchy. And that's what gives you kind of this awesome material that both absorbs energy and stretches. So that's what the repeats do, is because the silk's folding back on itself, or there's multiple proteins and they're lining up different parts. With the glue sequence, the protein molecule doesn't fold in the same way that the silks do. So it actually has all these little sugars tacked on that makes it sticky. And that part I didn't go into because that's a different part of the biology of the glue. So it doesn't actually have, the DNA itself doesn't have structure, but the DNA allows all of these sticky sugars to be stuck on. And so the more repeats you have, the more sticky stuff you can have stuck onto your protein molecule, essentially.
Okay, there's some more questions about the repeat nature. Does the repeat DNA translate into repeat sequences of amino acids?
Yes. So whatever the repetitive part of the DNA, that's directly translated consistently into amino acids. So if the DNA is repetitive, the amino acid sequence is going to be repetitive too.
And then for the genes that are extremely long and repetitive, do they express for anything similarly? Phenologically, can we see the evidence of repetitiveness?
Wait, say that again?
For genes that are extremely long and repetitive, do they express for anything similarly? Phenologically, can we see evidence of the repetitiveness?
Do they express for anything similarly?
You might want to you can unmute yourself and explain the question more if we're not getting it.
Is that listed in the chat? Is it kind of far?
It is. Here she comes. Oh, you're muted.
Yeah, I'm sorry. No, I mean, like anything that would have really long and repetitive genes, are they all going to be similar? Like, is the organism going to have some kind of similarity because their genes are long and repetitive? Or
Well, so okay, I think I see what you're saying. So there's lots of different kinds of repetitive DNA. So the very ends of your chromosomes, the DNA at the very ends are called centromeres. You've probably heard of centromeres. That's crazy, or sorry, telomeres. The telomeres at the end, that's crazy repetitive DNA. The center of your chromosomes, the centromeres, super crazy long repetitive DNA. There's things called microsatellites that are like they're really repetitive in different ways. So all organisms, we have them, everything has telomeres and centromeres, they all have repetitive DNA. So, and they all function differently. So I would say that the repetitiveness doesn't mean a whole lot and when it comes to relationships between organisms.
And now going 30,000 feet, how does global warming affect spiders and their webs? And how do you think that will affect our ecosystems?
That's a good question. Spiders like it warm, so I expect it'd probably be good for the spiders. Well, okay, I take that back. There is a spider that lives on the slopes of Mount Everest, and it catches like dead detritus that like gets blown up in the wind. So it's going to be really bad for those spiders. There are spiders that like colder climates. Trapdoor spiders in different places like it cold. There are tarantula species that like it cold. It's a funny anecdote, but there's, I'm in Charlotte now and they don't have these up in DC, but there's a spiny orb weaver that I used to only see in the Piedmont and like down on the Atlantic coast. And recently someone sent me a message saying, oh, like, what is this spider? And it was a friend from back home where I'm from upstate South Carolina near Clemson. And I was like, this isn't near Clemson, is it? Like, I've never seen one there. And she was like, oh yeah, this is in my backyard. I've seen them all over the place this year. So I, like, anecdotally, I think that it's already happening where some of these spiders are extending their ranges because it's getting a little bit warmer for them.
All right. Does anybody have any other questions?
yeah, I was wondering, do fishing spiders use glue and webs, or do they just drop themselves down?
They use silk for sure, but they don't use glue. So not all spiders have glue. Some like wandering spiders and wolf spiders and things, they don't have glue because they're not really making webs. And they have like a kind of stickier silk that they use to like stamp their silks and attach it to surfaces, but that's another kind of more solid silk. But they don't have the same kind of glue that I was talking about, like the aggregate glue. So they just don't have it at all.
Well, on behalf of the Natural History Society of Maryland and everybody here, I want to extend our heartfelt thanks to Dr. Stellwagen for sharing her knowledge and expertise with us. Now I look around, everybody's brain is bigger than we started, which is great. And now your job is to take this knowledge and share it with others who you come in contact with. I can't wait now to go out and take a closer look at spider webs and having this knowledge now, and I'm going to be looking at them. I might get a microscope out and I'm going to be looking for glue droplets. So it's going to give me a whole different perspective of spider webs when I look at them or when I come in contact with them as I'm hiking through the woods and they're all over my face.
Right.
Yep. So again, I hope that you will join us for another program. We have a whole vast array of subjects that are going to be covered that I talked about earlier. Join us on Facebook, Instagram, our website, our mailing list, our meetup group, so that you can stay involved. Join the organization, join a club, join the community of the curious that we are as the Natural History Society of Maryland. And have a great night, and everybody stay safe.
Awesome. Thanks, everybody. And to the little girl with Katie and Oliver's name, you're a future arachnologist. I can feel it. Feel it.
There's still so much to know. The more we learn, the more we realize, the more we have to learn. So
yeah, awesome. Thanks for having me, everyone.
Thank you.
Bronwyn, I wanted to stay after so we can save the chat.
Yeah, I'm going to pause the recording now.